name of the conference, yes, and like what it is all about.
This conference was held at the Janelia Research Campus in Ashburn, Virginia. Of this bootcamp was imaging across biological length scales. So that means you're going to look at something which is as small as a cell to something as big as an entire organism. And that is exactly what we did. So this was like a 2 week course, very intensive. So we started with imaging cells, and by the end of the course, we were actually imaging zebrafish. So it's like from a single cell to an entire organism. And the different microscopes that are available there, you get to actually use many of them.
Welcome, Dr. Saumitra, and thank you for giving this opportunity so that I can discuss along with all the audiences, especially the students who are working in the field of bioimaging and they want to have an exposure where a very specialized or focused workshop or a conference is being carried out. And if it is international conference, then we always understand the importance of such conferences where you get to see different types of high-end microscopes, learn about the techniques and all. And so today we will be discussing, or I'll be rather requesting you to share your recent experience. You participated in one of such conference recently. So what is this conference all about, and how did you came to know? So first of all, the name of the conference, yes, and like what it is all about.
Okay.
If you can give
so
some
thank
idea.
you, Dr. Somnath, for inviting me and letting me share this experience that I had in the last, I think it was what, 2 weeks back, almost 2 weeks back. Yeah. So this conference was held at the Janelia Research Campus in Ashburn, Virginia. And the name of the, actually it's not a conference, it was more like a boot camp. So the name of this boot camp was Imaging Across Biological Length Scales. So, I mean, if you go by the name, it says across biological length scales. So that means you're going to look at something which is as small as a cell to something as big as an entire organism. And that is exactly what we did. So over the course of, so this was like a 2 week course, very intensive. So we started with imaging cells, and by the end of the course, we were actually imaging zebrafish. So it's like from a single cell to an entire organism. And the different microscopes that are available there, you get to actually use many of them. And it is more like every particular experiment or every particular imaging modality needs a particular microscope. It is not going to be like, okay, you can use one microscope for everything. So that doesn't really happen. So I think one very important part of this course was to uh, understand or basically realize the fact that you always don't need a high-end microscope to image whatever you're looking at. I mean, it very much depends on your sample and the question, the biological question that you are asking. So sometimes a simple epifluorescent wide field microscope can give you a lot more information than, like, say, you are looking at a confocal or a super resolution microscope. So, I mean, of course, we did a lot of hands-on and we got a lot of exposure to different high-end microscopes. But I think the take-home message overall from the entire course was that you need to design your experiments, you need to understand what your question is, and accordingly choose your microscope. So that, I think, was the overall overarching, theme behind it. Yeah.
Okay. So it sounds very interesting because from a cellular level to a multicellular level and going to an organism model, the complexity of the sample type and also the requirement of various modes of imaging also varies. And it sounds really fascinating for me at least. Okay, but coming to a very basic question, if I am a student, how can I be informed about such workshops happening?
Okay, so I think one important thing is that it depends on your interest. So if you're interested in a particular thing, you will be looking at it. I mean, you will be searching, you will be mostly Googling it and trying to figure out, okay, what are the resources that are available to me in terms of like there's a lot of information available on the internet. You could always look at it. So I mean, I knew about Janelia from a long time back. I mean, since I was doing my postdoc, and Janelia was pretty much close to the NIH, so that's something that I already knew. So I had already subscribed to the newsletter. So every month or very frequently they keep on sending you emails telling like what are the different conferences or different workshops that are available. So I came to know it from the newsletter that was sent to me. But as a student, if you do not have that exposure, or if you do not know like what are the good training institutes, the best thing is to really like type into Google microscopy training programs or microscopy training courses, and I'm sure the internet will actually give you a lot of information, and you can actually individually visit those websites and obviously figure out. But as far as microscopy training is concerned, I think EMBL in Heidelberg, Germany is one place where they do a lot of microscopy training. Janelia is another place where they do a lot of training, and there are places like MBL, Marine Biological Laboratory, that's on the East Coast. I think it's in Massachusetts, somewhere in close to New York, somewhere. So these are places where you should actually look for. But for a start, obviously, you just Google and you will definitely get a head start, and then you can, of course, look for it.
Yeah, yeah. I think we have a similar workshop here in
Yeah, the Bangalore
NCBS,
microscopy
Bangalore.
course. Yes,
That is
yes.
the Bangalore. Yeah, yeah. So they also do it annually, and every year, no, there are a lot of
Okay.
students who apply for this. So it's just one of the best courses in
Yes, yes.
India, as I believe.
So I mean, this course that I attended was mostly like it was a bootcamp course, so it was mainly meant for people who are in core facility staff, scientists. But there were also some grad students, PhD students, but it was more focused towards training the core facility people. So the idea behind this entire bootcamp course was like you train the trainers. So you train the trainers, and the trainers can come and then train other people as well. So that was the idea, and this happens like once in 2 years. So, yeah. Okay, so once you are interested and you want to really apply, so they have a pretty simple form that needs to be filled up. Basically, the question is like, why do you want to apply to this course? So there has to be a very strong motive behind applying to this course. And another important thing is that what is the basic microscopy knowledge that you have, or in other words, what are the microscopes that you normally use? So the minimal expectation is that you at least are familiar with wide field microscope, confocal to some extent, and maybe at least one of the super resolution modalities. And your image processing part, you should be a little bit familiar with, say, Fiji or ImageJ. Uh, Better if you also are familiar with Imaris and any other high-end imaging software. But the idea is that you need to have a certain basic understanding of microscopy and image processing, so that when you go there, you're not like somebody who's totally new to the thing. Also, if it is important that you have a basic understanding of the optics, the physics behind microscopes, not in details, but at least the basic principles, the properties of light and how you're using light as a property to visualize your sample. So I think that is the basic thing that is required. But to answer your question, yes, you need to tell why you want to attend this course, how that course is going to be beneficial for you, what is it that you're going to learn from it and bring back with you, and how that is going to be helpful for the people, or if you are a scientist, like how is that going to help you train other students, specifically like as a core facility personnel. If you are learning something, how easily you're going to dispense that information to your students or to other people who are using the facility. So as long as you explain that properly, there are good chances that no, they'll select you. So it is very competitive, let me tell you. So this year there were 381 applications from around the globe, and they selected 24 of them. So that's quite that's quite it. Yeah.
Yeah, yeah, tough, tough. So in a nutshell, if I say that you have to have your
Yes.
SOP very strong, You need to have your current CV where it mentions that you are presently engaged into imaging process and you have a preliminary exposure of microscopy as well as
Yes.
certain platforms of image processing. That is the second criteria. And any other affiliations are also required. Like if I have to be affiliated with the institute, then only I can apply.
That was not really mentioned, but I think, yeah, that would be important. I mean, if you're a student or you are a core facility personnel, most likely you are associated with an institute. So yeah, that is like, it's kind of, it's understood. Yeah.
Okay. Now, my idea of asking this question is, do I need a recommendation letter from the supervisor I'm
No,
working
I did
with?
not need one. So I think this course does not require, but there are, I mean, there are other courses by other facilities where, yeah, you definitely need a letter of recommendation from your supervisor or from your mentor. So yeah.
Okay. So any quick advice that out of odd 380 400 applications, how I can make myself a most suitable candidate and my chances of getting selected for such workshops increases? Any tips or any
I think,
feedbacks
see,
from?
it's important that you convey to them how this training is going to be helpful for you and also the organization that you work for. There has to be an immediate what do you say, immediate dispensation of the knowledge. So you learn something and then you come back and you immediately share that information with others. So that, I think, is one of the most important criteria. Second thing is, of course, as I said, you need to have a basic understanding of microscopy. That should be evident from maybe your publications or not somewhere. So that is important. And it's important that they have a small questionnaire also. So they ask you like, so how often do you use a confocal, for example, in terms of hours per week? How often do we use a super resolution? Are you familiar with handling big data sets and all that? So it is important to answer that truthfully. I mean, not every answer needs to be yes. If you just think that, okay, if I answer yes to everything, my chance of getting selected is higher, that doesn't really happen. So you have to be honest with whatever you say, and yeah, you have to tell them like how it is going to be useful for you and how that is important for your career, and also how that is going to be useful for your institution. So as long as that is clear, I think, yeah, should work.
Right, right. So I think the prior preparations are what all you mentioned. Apart from
Um,
that, anything else
not,
in terms of specialized any
not
course
really.
module
You
which
should
we
just
need to
know the
complete
basics,
before?
and the course begins with a lot of basic lectures. So you can always, pick up from there and learn from there. And before you leave for this course, they will send you certain materials to, read through. So that really helps. But I mean, you just need to have a good basic understanding. That is it. I don't think there's anything more than that.
Okay, yeah, yeah. Okay, so from India, you are there.
No,
Anyone else
nobody
from
else. No
India
one else from
also
India.
participated? Okay, okay, great. Now, as we have understood our preparation part and then applying for the workshop and getting selected, so that part is done. If you can give me a overview about what this workshop was in terms of how many days this workshop was planned, and as per the schedule, what were the lectures, the hands-on, or it was entirely theoretical or entirely it was practical, how it was distributed or how it was organized to you? What is, can you share that entire
So this was
workshop?
for 2 weeks. We started from the 28th of July and we completed it on, I think, 9th of August. So it was like 2 weeks. And it was a very nice blend of lectures and hands on. So usually the morning sessions would be the lectures, and the afternoon you would spend time with the microscopes. So that is how it was arranged. And there used to be like you'd be explained a concept, you'd be explained the principle behind, say, how an airy scan works, and then immediately in the afternoon you would go and you would work on an airy scan based microscope. You would, and there used to be a very defined set of experiments. You used to get like hands out. So sorry, handouts. So the handouts would have like this is what we are expecting you to do. You will be given this XYZ sample, and you would need to image it. You have to decide what are the parameters that you are going to basically incorporate in your imaging setup, and why are you actually implementing that particular setup. Like, you want to image for 15 minutes, or you want to image it for half an hour. What is the, maybe the frame rate? If you're taking a video, what is the frame rate of the sample? What is the exposure? And what is the laser power that you are setting? uh, why are you exactly doing that? And then, so it is not just going there and doing the experiment, you get your data, and the next day as a group. So we were like 24 people, we were divided into 4 groups. So each group would have to come and give a presentation on what was the experiment all about, how did they perform the experiment, and then how did they analyze the data, and what is the data basically telling. So you make a hypothesis, you prove or disprove your hypothesis, and you say what are the problems that you face, what are the limitations of this method, what are the problems that you face. And then there used to be a overall discussion, like, okay, you could do this, or yeah, this was a good idea. So it's very like intense and very involved. It's not like you just go there and you spend some time with the microscope and okay. So they're not really teaching you how to handle a microscope. They are totally like trying to give you a proper mindset. Like, you need to have a proper, hypothesis. So I think most of the experiments were that way. And then there used to be lectures on image processing. So that would be like, the person would be actually explaining on the podium, and we all used to be with our laptops. So we already had the software downloaded, and there used to be a common database network basically. So we could just get the images from the network, and as the person is explaining, we would be actually working on our computers and following him. So that way you get a hands-on training on the processing of your samples as well. And then of course you apply that when you get your own data out of the microscope, you get to work on the processing part, and then of course you explain to the group. So that is how this whole thing was developed. Yeah.
Now, one thing I liked it, or it's very unusual in India, especially because here people are always told about protocols. You follow this protocol to do it. But imaging is one area where protocols or set norms
Yes.
doesn't always apply. You have to be intuitive, and you have to use your always,
Yeah.
depending on the samples you're using. So on that note, can you share one, sample type, and what all things you tried? Maybe some difficult
So
samples.
I think for the cellular imaging, I mean, the cellular imaging wasn't that very difficult in terms of the samples. We had cells on a dish, basically, stained with different dyes, and we were taking it at different channels. That wasn't really a problem. Another, I think, experiment that we did was, I think, a wound healing assay. So that was like the sample was left overnight, and we had to see how there was a scratch, and then the two sides of the tissues comes together, and no, the wound is basically healed. So that was something that so even that wasn't really challenging. I think the challenging part for me, I would say, was when we are doing organism scale imaging. So we use the light sheet microscope for imaging the entire zebrafish. So this, from the sample preparation to embedding the samples in agar, and then checking the orientation, making the light sheet in a proper orientation. I think those kind of experiments were slightly more challenging than the cells and other part. So yeah.
Yeah, as you mentioned about light sheet. So just it's out of inquisitiveness. What all different systems
So,
were there for your
so
use?
Janelia is known for its own set of microscopes, and yeah, I mean the person who got a Nobel Prize, Eric Bedzik. So his lattice light sheet is still lying there in Janelia, and we got to actually see that as well. So that's pretty much an open microscope. It doesn't look like a microscope that we are so much used to seeing, totally housed. It's pretty much open, and you can just see the light, the lasers going in and out. So they have a lot of their own microscopes. We used one from Zeiss. I think it was the Light Sheet Z1, and they also had the Light Sheet 7, I think. So yeah, those two systems were from Zeiss. There was one from Bruker, if I'm not mistaken. I don't know what that was called. Um, I don't actually recall the name, but that was one from Bruker. And then, of course, there were a couple of microscopes which were made at Janelia itself, like the SimView and the others. So yeah, so that was more of it.
Okay, okay. You also mentioned about Eriscan, and there were any systems from Nikon and Olympus?
Ariscan, we used the one from Zeiss. We did use the Nikon system, but I think that was the spinning disc that we used from Nikon. Yeah. Uh, I don't really recall that. Mostly, we had systems from Olympus and Zeiss, and the homemade ones. The stead that we used was from Abirier. Yeah, I think that was quite it. Yeah.
Wow. Oh, I think these are the major. Why I mentioned this, because
Yeah.
these are the major companies, right? Who are global manufacturers of microscopy, and there might be certain pluses and minuses, or I would say there might be certain differences. They might be very subtle. The platforms are all should be same, but the quality and the
Yeah.
ease of handling of the images is something
Yeah.
which you should be, yeah, trained on. So my idea was to ask this question just to know what exposure one can expect when they go to such workshops and see on different systems, hands on, or using them.
Yeah, I mean, see, every system has its own pros and cons. So they are basically designed for a particular reason. So I would say that nothing, no system is good or no system is bad. I mean, it all depends on what you're actually looking at and how optimally you can utilize the microscope. And one good thing with Janelia is that they have their own scientists, physicists, and biologists who work together, and they make their own microscopes. And once their prototype is basically ready, they invite this commercial people from Zeiss or from Olympus, they come, they look at it, and if they feel that the market is good for this kind of a system, they take the system from there and they try to commercialize it. So that is basically how the light sheet actually came into the market. So Eric Bedzik has made his light sheet. Zeiss came over, they had a look at it, they felt that the market is going to be, I mean, it's going to be a good place for the market. So they built their own this thing. Of course, the basic idea is going to be the one that was made in Janelia, and then of course they kind of customized it for different types of samples. So that is one thing that Janelia, I think even EMBL does that a lot. So they have their own prototypes, and once they think it is ready, these commercial companies, they come over, they see it, and then they take it from there. And that's how, like, I think that's how many of the microscopes right now in the market are actually there because of kind of.
Yeah, I think this collaboration of industry and academia will also help them to
Yeah,
test their models and make it more
yeah,
future
that
ready.
is very true. And one more thing with Janelia is, I think, I don't know how many people would have heard about Thor Labs. So Thor Labs is one company which makes a lot of this microscopy stuff. The minute stuff, like sometimes you need some holders, or sometimes you need some, basic small stuff. Yeah, and Thor Labs is very close to the Janelia campus. So it's like scientists just walk in from Janelia. They just walk into the Thor Labs lab and just look at the certain things. So I mean, and whenever Thor Labs comes out with some equipment, they usually send it over to Janelia. They sit down there. I mean, they get to really test their samples for free. I mean, so that's really like, so that's the beauty of it. And it's also important that you have your own, like any institute, if you have your own, like a factory kind of a place where there are small holders or something that you can actually manufacture according to your needs, that really helps. And I think TIFR has that. TIFR has its own place where there are certain small fixtures, or stuff that is needed. Like sometimes what it does is, every commercial company is not going to give you holders for everything. There could be something that you want to just make your own thing, like you want to image, for example, a Drosophila or a C. elegans, but you just need something very specific that no that is required to fit in your sample. And I mean, Zeiss is not going to make that for you, or no, Olympus is not going to make that for you. So you will have to do it yourself. And if you have a factory wherein you can go and tell them the dimensions of the holder, or no, tell them the specifications, then they can come out of it. So this kind of a setup is very important. Yeah.
Right, right. No, I totally agree, because I've seen in the Thor Labs is one go-to company where you are a person where you need modifications in your microscope, and those modifications, or as you said, no, like if I need a holder, if I, so I remember some time back we were looking for two illumination from the back port of the microscope, and we wanted to integrate both the illuminations, one like may not be simultaneous but one after another, but the adapter was compatible for only one
Okay.
illumination hub. So Thorlab was the only company which was providing the laser line or the path. It was bifurcating into two, where two illumination sources can be integrated separately. Depending on your requirement, you illuminate one light source
Yeah.
and then you use it. So these solutions I think nobody else offers till date. I think
Yeah.
Thorlabs does it pretty well.
So that really helps when you're close by. You have a lot of networking happening, and it's easy to convey what you're looking at. So I think that really, yeah, is an advantage for places like Janelia or EMBL. So that really helps.
True, true. Okay. So, as I wanted to understand, the curriculum which you have beautifully explained, I think it's really wonderful to see under one roof. You see such different types of instruments, in terms of complexity as well as in terms of modalities also, and able to run few experiments from cellular level to like even for the light sheet going to the organism level also. But if you want to say, like, if I am a enthusiast and I just want to pursue in future as a imaging as my career, what would be your message
So
to them?
imaging as a career, I think it actually depends on. I think. See, the thing is, you get exposed to imaging probably when you are starting your PhD. At the master's level, I don't really think you do that much of an imaging, unless, of course, you are doing your MSc from a research institute, where research is a very important part of your curriculum, pretty much like ICERs or IITs, maybe even AIIMS for that matter. I think where you have to submit your MSc thesis, and that's where you might actually get an exposure, but it may not be that very intense. So probably the first time that you are going to really spend a lot of time on the microscope is going to be during your PhD. So that would be a time when you actually decide, like, if you really like imaging, if you like, spending time in a dark room at 18 degrees or 22 degrees, and no, really love processing those images, and you're really fascinated by it. So probably your PhD is going to be a time when you take that decision. And I mean, it all depends on, like, what you really want to do. I mean, in terms of career as prospects, you could have, I mean, you could do your PhD, then move on to do a postdoc where you can continue to explore and improvise on your imaging knowledge, get exposed to more microscopes, more like high-end super resolution and stuff. And I think it's at a postdoc level you can kind of decide if you really want to take that as a career. Now, when I say take that as a career, it is not necessarily academia. You could also move to industry. I mean, there are multiple imaging companies like who make microscopes. So you could actually join them. That could be one option. Or in terms of academia, uh, you could join a core facility and work there as a scientist or a research analyst. That could be one option. Or if you want to be a PI and have your own lab, uh, provided you are mostly doing cell biology kind of work, where maybe say 80-85% of your research basically is dependent on the microscope. So these are a few of the career options that I can think of. So if that is what interests you, you have to, like, kind of go in that direction, because I think in the Indian curriculum you don't really have microscopy as a very specialized subject. You're going to probably have some explanation of microscopy if you're doing cell biology, or if you're studying anatomy, maybe microscopy is going to be one chapter in it, or I don't know, like, it's going to be very basic. If you're doing your PhD, maybe in your coursework you might have microscopy as a subject. So that would be one place where you would get a lot of microscopy related information and knowledge. But otherwise, I would say that you have to read the microscopy books on your own. And then, of course, if you are also doing your experiments, you try to like apply whatever you've learned. You try to basically apply that in your work. So that's kind of the thing. So yeah, I mean, to give a very short answer to your question, uh, there is nothing like there is no straightforward route to yeah to understand the microscopy. It's more like you are like you learn it on the go, and that, yeah, I mean it depends. Like once you reach a career stage where you have to move from being a postdoc to the next position, that's where you can actually take that call whether you want to go towards one of those companies or you want to continue in an academic institution and take care of the imaging facility. So yeah, that's totally like your call basically at the end of the day.
Yeah, true, true. So, lastly, what is your take-home message, after attending such workshops?
I think what I would say is there is a lot of information available, and there are a lot of people who are very good at microscopy and whom you can learn from. But the problem is we are not aware of many of the things, and that is, I think, one major problem. And this is also something that we discussed. So the information dissemination is very important. And I think, especially in the context of India, I think we just have this Bangalore microscopy course, which is, I think, the one microscopy course that actually happens. Uh, Fortunately, we have this India bioimaging network that is, I think, kind of now building up. We have a meeting in ICER Pune that is going to happen in December, and we are going to, work out the modalities of how we can actually make microscopy training and microscopy education more accessible to students and researchers in India. So that is one of the directives that we are all working on as a team. So I think what I would actually say is it is important that we start many of these training programs in India, because I know it's not easy for everybody to travel abroad. Uh, there are a lot of complications from even applying and from getting your visas and traveling all the way. There are a lot of expenses, so it's not easy to even get funding many a times to attend these trainings. So it is important that you have people in the country who can actually train you. So yeah, I mean, once we have this network of institutions and network of people, I think it is going to be very beneficial for the young students and the researchers to get the access. So I think right now the challenge, of course, from an Indian perspective, would be to have a lot more of these trainings, not just have one or two trainings a year. We should have multiple levels of trainings, from not just from just hands-on training on imaging, but also I think image processing, because image processing is equally important. And not everyone is going to have access to a confocal or a super resolution system, but sometimes even a wide field image, if analyzed and processed properly, will tell you a lot of things. So yeah, so I think that should be the priority right now, that we need to really increase and improve the training in India, so that a lot of students are benefited from it.
Yeah, sure. Thank you. Thank you. That was really very insightful. And whoever might see this, they might get a lot of, at least they can plan their roadmap. That if they're planning, yeah, so if they're planning to attend such workshops or boot camps, I think they can plan it much better, and the expectations out of attending such boot camps can also be set, right? So thank you, Dr. Saumitra, for your wonderful experience sharing, and whoever will be seeing this, I think they'll be
I hope
immensely
so.
benefited out of it, right? No, I know so, because for a student, it's always, life is, has a lot of challenges in terms of fidgeting out for different informations, which informations are there in these days, but to filter out the correct information and use them in our life is something which is challenging these days. On that scenario or on that platform, I think this discussion today what we had would
Yeah,
literally
I
help
think the
them.
other important thing is that I mean we are all busy in our lives, so we probably are not able to give that much time to students. I think that is another problem. So I think it is also our social and moral responsibility, I would say, that no, we give time. We have to find out platforms. We have to find out ways where the students can basically access facilities, access the scientists in the facilities, and get to talk to them, and learn more about image analysis, image processing, or even like using the right microscopes of their experiments. I think that is also an important thing that one should actually look at. And there has to be a collaboration between the institutes and the industry, because obviously if we are not collaborating with the makers of the microscopes, it's not going to be easy for us. So it is always a two-way communication, and it cannot be a monologue. So I think that is another thing that we really need to work on. Yeah.
Sure, sure, sure. Thank you for your time. Thank you for your wonderful inputs and feedbacks. I think whoever will be watching this, they can no at least no prepare for the next year. Get updated with what all upcoming webinars or conferences specialized related to bioimaging or light microscopy is coming up, and they might, or I would say they should, attend one such conferences during their PhDs and postdocs. That is highly recommended.
Thank
Yeah.
you.
Thank you. Thank you.